Hello, today we are going to know about what to do when in exam we are given culture of bacterial colony which has grown after overnight incubation on a plate which may be nutrient agar, which may be blood agar, which may be maconchi agar, which may be chocolate agar, or a broth which is given in liquid culture media. So first what we have to do from that culture media, we have to take a look of colony and then we have to place it on a slide and do a staining process that is called gram stain. First I'll take it in a slide. After marking it, then we will put the colony and make a smear. And then what to do? After making smear, we'll do gram staining. In gram staining, what are the steps? These are the chronology of the steps. First is crystal violet. For 1 minute, we'll put it. This is the primary state. It will color it, and then I'll give a gram iodine, which I'll keep for 1 minute, and then again I'll wash it. It will act as mordant, and then the third step, decolorizer, which has to be done for 10 seconds only, which can be acetone or alcohol. And then after washing it, then we'll put the counter stain as saffron in for 30 seconds, mind it, and then wash it, and you observe under 10x of microscope followed by 100x, and you get either purple colored cockeye shaped colonies, or you get pink colored gram negative bacilli or cocci. So once we identify the shape and morphology of the gram stain that we see under microscope as 100x, we can see gram positive cocci. Gram positive cocci can be arranged, they can be arranged in groups, or they can be arranged in pattern of two diplococci, or they can be arranged in chains and pairs. So from where we get the groups, then we know it is in cluster. Gram positive cocci in cluster. We can think it of as a genus called Staphylococcus species. But when we go for diplococci and chains or pairs, we'll do a test called catalase. By catalase test, if it is positive, then you know the all organisms belong to a family called Micrococcaceae, where will difference between Micrococcus and Staphylococcus. And this can be done on the basis of a test called modified oxidase test. And after that will followed by tests like coagulase test. But once catalase is negative, then we have to group it under Streptococcus. And if we see gram negative bacilli, then first step what we have to do is oxidase and catalase, and followed by motility. So we have to know like that if catalase is positive and oxidase is negative and the organism is non-motile, then, and on Mekong agar if we see lactose fermenter, then we'll think of it as Klebsiella. If it is non-lactose fermenter and catalase positive, then we can think of Shigella.